Welcome, everybody, to our March edition of the Gene Delivery Seminar Series. We are very, very, very excited to have the speaker today, Skye. Dr. Skye Storrie carried out her undergraduate studies at the University of Glasgow, studying virology and completing a Master of Research following a year-long placement at the Morden Research Institute in Edinburgh. She then successfully blagged her way onto the Wellcome Trust Infection, Immunity, and Inflammation program at the University of Cambridge, where she undertook research rotations ranging from tuberculosis to a novel ant virus. Skye completed her PhD researching Zika virus, investigating how a protein named after a Japanese samurai, Musashi 1, influences viral pathology in neonates. After her PhD, she spent several years traveling Europe in a camper van, briefly masquerading as a mountain runner before joining Vector Builder as a scientific product manager. She now combines her passion for science and education to create scientific content and coordinate product launches with particular focus on lentivirus and AAV. We are very excited to have you, Skye, and whenever you're ready, take it away.
Perfect. Yeah, thank you so much, Sanitha, for the introduction. Yeah, and I'm really excited today to be here and talk to you guys all about lentivirus. So, this talk will be split into three main parts. So first, I'll do a pretty thorough deep dive into the background of lentivirus, just because we have quite a broadly academic audience, and some of you may have never worked with lentivirus before. So we'll cover the history of lentivirus, also how to use this natural system as a potent gene delivery tool, when you'd want to choose lentivirus over other viral and also non-viral delivery systems, and sort of the main applications that lentivirus is commonly used for. The second part of our talk will really be focusing on vector design. So I'll really be highlighting the importance of a rational vector design approach from the very beginning, and also sharing some useful tips and tricks that we've picked up along the way of combined decades of cloning these vectors and using lentivirus to help you, regardless of whether that application is for research or it's for clinical development. Thirdly, I will delve into packaging optimization. So how to optimize your packaging protocol to manufacture lentivirus that is really high titer and high quality, so it will be effective for your desired application. So first, let's dive into a bit of a background on lentivirus. So how did lentivirus go from being a viral pathogen to a powerful gene delivery tool? So to answer this question, let's take a quick step back in time to 1935. So lentiviruses were first discovered as a slow-acting disease in livestock, causing both pulmonary and central nervous system disease in sheep. And at this point, it was really only veterinarians that were really interested in lentiviruses because of the huge economic losses that this was causing to sheep farmers. It wasn't really until the 1980s that there was a bit more of a focus on lentivirus research, and this was due to the emergence of one of the most famous lentiviruses of all time, HIV, which was responsible for causing the AIDS pandemic. And whilst research efforts were more focused on lentivirus, this helped us gain a much deeper understanding on things like genomic organization, as well as the function of viral proteins. And this is what really helped us to take this system and harness it for gene delivery. And now lentivirus is one of actually the most popular recombinant viruses used in research. So this is the basic structure of a lentiviral virion, with its double-stranded RNA genome at the core, surrounded by the capsid, the nucleocapsid, capsid, matrix, and also these outer envelope proteins. In the center of the virion, we also have some viral enzymes, which are important during different stages of the viral life cycle, and we'll go into this again a little bit later. And if we zoom into this genomic RNA and take a look, this is the structure. So we have the viral proteins encoded, and these are between the 5' and 3' long terminal repeats, which are also known as LTRs. And these structures are both identical in sequence, and they're really important for transcription of the viral genome. There are three main open reading frames encoded within the genome, the first of which is GAG, and this encodes our structural proteins, so again, our nucleocapsid, capsid, and matrix. And then the next one is the pol gene, and this encodes our viral enzymes, which, as I said, are important for different stages of the viral life cycle. We also have the ENV gene, which encodes the envelope proteins, and these are really important for binding to host cell receptors to allow the virus to gain entry into host cells and cause infection. We also have regulatory proteins, so TAT and REV. And as I'll go into a bit later when we look at the viral life cycle in the next slide, TAT is important for upregulating transcription, while REV is important for exporting the viral RNA out into the cytoplasm so that it's translated into viral proteins. We also have a number of accessory proteins shown here in light pink, and these are mainly important in a host environment, so for things like immune evasion. So I'm a virologist, so I'm a little bit obsessed with viral life cycles, and I'd like to take you through the wild-type lentivirus life cycle. So it starts with binding of the lentivirus via its envelope glycoproteins to the host cell receptor, which for HIV is CD4. The host and cell membranes then fuse together, releasing the capsid into the cytoplasm. And in the sort of early model of HIV infection, uncoating and release of the RNA into the cytoplasm occurred quite early on, and then you have reverse transcription of this viral RNA into DNA. But more recent models suggest that uncoating actually likely occurs at the nuclear membrane or even inside the nucleus itself, and the reverse transcription is actually occurring within this capsid structure. Once we have our viral DNA, it is then integrated into the host genome and acts as a template for transcription. So we get more copies of viral RNA, which are then spliced and make their way out to the cytoplasm for the first round of translation of viral proteins, generating TAT and REV. And like I previously mentioned, TAT is important for upregulating transcription, so it makes its way back into the nucleus to perform this role, and we get the production of even more copies of viral RNA. REV then makes its way into the nucleus as well and binds to this full-length RNA, exports it out into the cytoplasm, and then we get our next round of translation of viral proteins, generating the envelope, as well as the GAG-POL precursor and GAG proteins, and these encode all the structural proteins. The envelope protein makes its way up to the host cell membrane and embeds itself within it. And then assembly occurs where the viral RNA genome, as well as these structural proteins, make their way up to the host cell membrane, where they become encapsulated by the host cell membrane, embedded with these viral envelope glycoproteins. This then kind of pinches off from the cell, and we have our immature virion, and then the structural proteins mature via cleavage of these precursor proteins, and these then rearrange themselves to form the final virion structure. And this virion is now fully infectious and can go on to infect the next target cell. So how do we take this natural system and harness it to deliver our specific gene of interest, or if we want to carry out RNA knockdown, shRNA, or if we want to do CRISPR, then gRNA. So to do this, we replace the viral genome with our gene of interest, driven by a promoter of our choosing. And we insert this between the 5' and 3' LTRs, because, as I said, these are really important for transcription. We also want to keep the packaging signal and the rev responsive element as well, because again, this is really important for packaging. So there have actually been several generations of packaging system developed in order to generate recombinant lentivirus. I'm not going to go into too much details about the first generation system. I'm going to kind of skip over it today. But if you're interested and you would like more information on this, soon we should be releasing a lentivirus sort of introductory video that will go into much more details on this. So please check that out. But for now, I'm going to focus on the second and third generation system. So for the second generation system, we take this cassette, which has our gene of interest, and we encode it in a plasmid known as the transfer plasmid. Then the genes that we require for generating novel virions, we will split between two plasmids. We don't encode any of the accessory proteins because these aren't really required. Instead, we just have GAG and POL, as well as TAT and REV encoded in our packaging plasmid, and then the envelope encoded in our envelope plasmid. And then if we transfect a packaging cell line, such as HEC293Ts, with these three plasmids, we will generate recombinant virions, which will encode our gene of interest. But now a lot of people are moving more towards the third generation system. And this is because there are a number of safety concerns associated with the second generation system, including the fact that recombination can actually lead to the generation of replication competent viruses. And also because the 5' and 3' long terminal repeats do actually have enhancer promoter activity. And following integration of the viral genome into the host genome, if it inserts near proto-oncogenes, this can actually lead to cancer. So there are several modifications that have been made to the third generation system to try and enhance safety. Probably the most important of which is actually the self-inactivation mechanism, which is encoded in the 3' LTR as a deletion, which is then copied into the 5' LTR during reverse transcription. And this actually disrupts that enhancer promoter activity that I was just talking about. So you don't have to worry so much about sort of triggering nearby proto-oncogenes. Because we've disrupted the promoter activity of the LTR, and normally the TAP protein will bind to the LTR to drive transcription, we can actually just remove the TAP protein completely from the system. And to ensure that we still drive transcription, we just include an upstream promoter. So in this case, we've included CMV. And by removing this extra protein, we're also reducing the risk of recombination and therefore, again, reducing the risks of generating replication competent virus. Another feature which enhances safety is that the viral proteins are split across 3 plasmids instead of 2. And again, this also will reduce the risk of recombination. So overall, the third generation system is much safer. And for example, in terms of clinical development, these are really important modifications that have meant that it has been able to be used for therapeutics development. And then again, we just transfect our packaging cells with these 4 plasmids and we get our lentivirus, which will express our desired transgene. And so if we take a look at what the recombinant virus life cycle looks like compared to wild-type lentivirus, the beginning very closely mimics the lentivirus, wild-type lentivirus life cycle, where we have entry and uncoating and release of our viral RNA, which will then be reverse transcribed into DNA. But this is kind of where the main differences lie. So once we have integration of our RNA of our DNA into the host genome, this will act as a template for transcription of more copies of our transgene RNA because it doesn't encode any of the viral proteins. So we won't get the production of novel virions, we will just get translation and expression of our transgene. So why choose lentivirus? There are lots of different gene delivery systems out there that are available. But first, let's look at why you would maybe opt for viral vector systems over non-viral vectors. So non-viral vectors include things like regular plasmid, transposon, or RNA. And the main advantages of these types of systems is that they're pretty easy to produce and generally safer than viral vectors. However, the main sort of drawbacks of these systems is that it is more complex to deliver. So often you need to use physical methods of delivery, such as electroporation or microinjection, and this can actually be quite damaging to the cells. Additionally, it can be quite difficult to achieve targeted delivery, and it can be quite low efficiency. If we look at our viral vectors, so some common ones that are used are, of course, lentivirus, as well as AAV and adenovirus. The sort of main advantages of this is that you're utilizing this natural system and you can sort of harness the infectivity of the virus, so it's a bit easier to deliver. You can also sort of harness some natural aspects of the virus's targeting capabilities to target specific cells and tissues that you want to. But of course, the main drawback of this, as we just talked about, is you've got to package your virus. So this involves cloning all your different plasmid constructs and then packaging and producing your virus, as well as QC and purification, all these things. So it is a bit more complicated production. Additionally, these are viruses which are associated with higher safety risks. So, for example, they can cause high immunogenicity and toxicity, and also for lentivirus, where it integrates, you can get oncogenesis. So, how does lentivirus compare to other popular viral delivery systems? So if you want stable long-term expression of your gene, then lentivirus and MMLV are both good choices because they integrate into the host genome. If you want more transient expression, then you can use viruses such as AAV and adenovirus because these don't integrate. Interestingly, for AAV, if you're transducing non-dividing cells, it can actually hang around for quite a long time because it exists as this very stable episome, unlike adenovirus, which has a linear genome and tends to get degraded faster. In terms of tropism, so targeting different cells and tissues, enveloped viruses like lentivirus and MMLV are good options. They're quite flexible, whereas viruses like AAV and adenovirus have this more rigid capsid structure. And if you start messing around with this, it can disrupt virion assembly. For envelope viruses, you can generally easily switch out the viral envelope glycoproteins for envelope glycoproteins from other viruses. There are some cases where lentivirus may not be your best option. So although it has a decent cargo capacity of around 6.4 kbs, which does surpass MMLV as well as AAV, if you want to deliver larger transgenes, then an option such as adenovirus may be better, which can accommodate up to 33 kilobases, depending on the type of adenovirus you use. And if you do start exceeding your cargo capacity of lentivirus, this can affect packaging efficiency. So this is an important consideration to make. In terms of applications, if you want to do sort of in vivo experiments or clinical applications, then AAV is generally a widely used vector. And this is because it has low immunogenicity compared to, for example, adenovirus, as well as low pathogenicity. But if you want to carry out in vitro experiments or ex vivo modifications, then lentivirus is a really popular choice. And if we take a look at some of the most commonly used applications for lentivirus, stable cell line generation is one, and this is because lentivirus has really high transduction efficiencies. Also, because you can pseudotype lentivirus, it means you can target a broad range of different cells. Additionally, you have that long-term expression because of integration into the host genome. Also, when you want to transduce a large number of cells, again at high efficiency, for something like library screening, where you might want to identify specific genes involved in certain pathways, then again, lentivirus is also a very popular option. For therapeutics development, lentivirus has become incredibly popular for ex vivo cell therapy, and this works by extracting patient cells, modifying them with lentivirus, expanding them, and then transfusing them back into the patient. And this has proven highly effective for cancer therapies. So, for example, CAR T, where you modify patient T cells with lentivirus so that they express the CAR receptor and then they can effectively, specifically target cancer cells. And although at the moment it's not quite there yet due to targeting and safety concerns, the future of in vivo cell therapy does look quite bright for lentivirus. There are lots of preclinical trials ongoing at the moment to sort of overcome these major obstacles. So now we've kind of delved into the background of lentivirus, and let's focus on vector design. So I just really want to highlight the scale of the problem and the fact that vector design mistakes are very costly. So regardless of whether you're doing foundational research or clinical development, if your vector is not well designed from the beginning, this can just lead to lots of wasted time and wasted resources where you essentially get so far along your experimental pipeline, and then you have to go all the way back to the beginning because of some little mistake. And interestingly, 90% of CGT clinical trials fail. So cell and gene therapy trials actually fail. And this is due to poor vector design as well as poor clinical performance. And this can be avoided using a rational vector design approach from the very beginning. And we feel like we really highlighted the scale of this problem in a paper we published all about plasmid errors across the globe. And we took thousands of plasmids from academic as well as industry partners, and we sequenced them to look for mutations, as well as carried out restriction enzyme digest to look for mutations. And we found that actually a whopping 15% contained significant design errors that could significantly impact function. And I think we've all done it. We get plasmids from collaborators or repositories, or we have that plasmid that's sort of been in the lab freezer for about 3 decades, and we just take it and go ahead and start using it in our experiments. But this really highlights the importance of making sure that what you're starting with is what you think it is and as well designed as well. So we believe that the solution to this is to employ a rational vector design approach, where all components of the vector are carefully considered from the very beginning. So this includes everything from the promoter to your gene of interest, whether you want to include a regulatory element or not, if you want to include a marker gene for selection, and also which antibiotic you want to use for selection. And the basis of this is that you really want to keep the endpoint in mind. So whether this will be used for foundational research or whether it will be used for clinical development. So first, let's start off with promoter. So the promoter is really vital to your construct because this is going to drive appropriate expression levels in the right place at the right time. And depending on whether you want ubiquitous expression or you want more regulated expression, there are lots of different promoters to choose from. So let's start with ubiquitous expression. So ubiquitous promoters are very commonly used, and that's because they generally work in lots of different cell types and they induce really high levels of expression. However, we don't always want super strong levels of expression. So for example, maybe you sort of want to replicate more physiologically relevant sort of levels, or maybe you want to express a toxic gene, so you don't want levels of expression to be too high. And in this case, other promoters may be more appropriate, like a ubiquitin promoter or PGK promoter. Additionally, promoter activity really varies by cell type. So here you can see that our CMV promoter is inducing really high levels of expression, but then in embryonic stem cells, the CMV promoter is actually prone to silencing. So you really need to consider, for your specific experiment, the cell types you're using when you select your promoter. Additionally, promoters can really affect manufacturability. So we've found from our internal data that the CAD promoter is actually associated with low lentiviral titers. So if you want to use the CAD promoter, you have to keep this in mind, because if you need really high titers for whatever application you're using the lentivirus for, then again, this might not be appropriate. Additionally, a common mistake we see repeated quite a lot that is a very important consideration is that you should not have repeat sequences in lentiviral vectors. And this is because these sequences can recombine and also this leads to truncation or even deletion of parts of your gene of interest. And this also applies to the promoters you're using. So you should never use the same promoter twice. And if we take a look at some of our internal data from this, we designed constructs where we have two separate fluorescent proteins driven by two separate promoters. In the top row, we've used the same promoters. So you can see here for A that this has actually led to recombination. And if we look at the bottom panel, which is our EGFP, so our upstream ORF, we've basically completely lost expression. And then if we look here, we do seem to maintain decent levels of expression of mCherry. But here, where we should be seeing co-expression of both EGFP and mCherry, we only have 11%. So you can see we've basically just completely lost expression of our upstream ORF, which is really not great. Then if we take a look at F, where we just have a tiny amount of overlap in sequence, we still actually lose expression of our upstream ORF. So again, down here we have about 2%, and we're only seeing 17% co-expression. So what do we recommend? We recommend using two separate promoters. So for example, you could use EF1A and CMV, and we get a nice level of co-expression at around 89%. So there are other types of promoters you can use if you want a little bit more control over your expression. So you want to control when and where your gene is expressed. So the first of which are tissue-specific promoters. So these will only activate expression of your gene of interest in a specific target cell or tissue. Important thing to note about tissue-specific promoters is that they generally do have weaker expression than ubiquitous promoters. So if high levels of expression are required for your experimental setup, then again, this may not be good for you. For temporal controls, for controlling when your gene is expressed, you can use inducible systems. So for example, the tetracycline inducible system. And briefly, there are two main types of systems. So one system where your gene of interest is switched off in response to treatment with a drug, such as tetracycline or one of its analogues like doxycycline. And this is known as the TET-off system. But the more popular system is this TET-on system, where gene expression is turned on in response to doxycycline treatment. And basically this works through having a number of constructs. So the first of which is your gene of interest driven by this tetracycline response element or tree promoter. And the other of which is your TET regulatory proteins. So in this case, a transcriptional activator, which is RTTA. And then when you add doxycycline, it binds to this transcriptional activator, which then induces it to bind to the tree promoter and drive expression of your transgene. And although this kind of works on its own, there can be some levels of what we call leaky expression, where even in absence of drug treatment, so in absence of doxycycline, we still get a little bit of expression of our ORF. And obviously, this is not great. You want inducible expression for a reason. So this could really affect your experiment. So to overcome this, you can also encode a transcriptional silencer, which basically will actively inhibit ORF expression in the absence of doxycycline. And if we take a look at some of our data where we're just using our transcriptional activators, so the top two, you can see that we do get a reasonable level of leaky expression. But then when we include both our transcriptional activator and our transcriptional repressor, we practically abolish leaky expression. Another important thing for lentiviral systems to consider is that we found that all-in-one systems where you express both your TET regulatory proteins and your ORF under a tree promoter from one vector actually doesn't work particularly well. So we do not recommend this. Instead, we recommend that you use either two separate lentiviruses, so one that expresses your regulatory proteins and one that expresses your ORF under the tree promoter. Alternatively, you could use lentivirus to generate stable cells that express these regulatory proteins, and then you can test lots of different constructs under the tree promoter and save yourself a bit of time in the long run. So what about if you want to express RNA? So you might want to express shRNA if you're carrying out knockdown experiments, or gRNA if you're carrying out CRISPR. And for this, you want to use an RNA polymerase 3 promoter, such as U6 or H1 for small RNA transcription. U6 is generally more potent than H1, so again, depending on what you want to achieve, that's important to keep in mind. For shRNA, you can also use miRNA scaffold, as shown here, such as MIR30. And the main advantage of this is that you can actually include whichever promoter you want upstream to drive expression of the shRNA. And like we just talked about, this means you could maybe use an inducible promoter, or you could even use a tissue-specific promoter. Additionally, MIR30 is great because you can, if you want to express multiple shRNAs, you can also do that as well. And here's some of our own data where we're comparing shRNA that's driven by U6 versus MIR30 and scaffold with a CMV promoter. And you can see that for U6, we achieve markedly greater shRNA knockdown efficiencies at 72% with our U6 promoter, which is much better compared to our MIR30 scaffold, where you can see that it's 60% reduction. And even when we use multiple shRNAs, our U6 shRNA still outperforms. So now let's look at the open reading frame. So some important considerations include cargo size. Like we previously mentioned, it should be around 6.4 kilobases. And this includes everything sort of in your expression cassette. So your promoter, your ORF, as well as any regulatory elements like poly-A or WPRE. So this is really important to consider. You also can do sequence composition optimization. So things like you might want to reduce GC content, you might want to do codon optimizations. And also the arrangement of your ORFs is really important. And all these optimizations can really significantly enhance expression. So lentivirus is appropriate for expression of multiple components. So for example, for CRISPR, lentivirus works really well for co-expression of gRNA as well as Cas9 from a single construct. However, as we just talked about, for some cases, like the TET system, multiple vectors are required and it doesn't work well to express all the components from a single vector. Another option is if you want to have lots of different ORFs being expressed from a single promoter, is that you can use these 2A linkers, which induce ribosome skipping. So during translation, you will get translation of the 3 separate proteins. And if we look at this a bit further, if you want to express multiple different ORFs using this method, it's really important to consider the order of the different ORFs. So the general rule of thumb is that the ORF that is closest to the promoter will be expressed at the highest levels, although this is not always the case. So in this case for gene C, you can see that at position 2, it's generally expressed at higher levels, which leaves position 1 open to A or B. And in this case, B is expressed much better in position 1. So you can only hope that gene A, you don't need too high levels of it, and it can be expressed at position 3. But again, I just want to really highlight the importance of testing this for your individual system, because this will vary wildly depending on the different types of genes that you are trying to express. And again, what we really recommend is to express your most important gene first to make sure that you're definitely getting good expression levels. So for marker optimization, appropriate marker inclusion, so something like fluorescent reporter or antibiotic resistance marker, can facilitate both visualization and selection. So if you can, we recommend using a dual marker design, so using both a fluorescent reporter as well as an antibiotic, just because it gives you a bit more flexibility in selection. If you want to use different fluorescent proteins, like a combination of different fluorescent proteins, you just have to be a bit careful that your signal does not cross over. So different combinations should be considered. So generally, green fluorescents like EGFP and mCherry work well together, and you can also throw in some blue fluorescents like tag BFP. But if you want to know more about all the different types of fluorescent proteins you could possibly use for different applications and all the new and upcoming shiny, exciting ones, I'd really recommend that you scan this barcode, and that will take you to another gene delivery seminar series, which is all about fluorescent reporters. Again, like I said, depending on how you want your markers expressed, you need to think about the order. So generally, we recommend that you would put your fluorescent reporter upstream so that you definitely get good expression levels of this, and then put your antibiotic resistance marker downstream. And this is because you only really need a really small amount of expression of your antibiotic resistance marker to allow antibiotic selection. Additionally, you need to optimize this depending on the specific cell type you want to use. So 293 T cells are really neomycin resistant. So neomycin resistance is not an option for these cells. Also, how you link your markers is surprisingly really important. So here we've linked our markers either by direct fusion, or we've used a 2A linker. And you can see, depending on the type of antibiotic that we've used, we actually see quite a marked difference. So for EGFP Puro, using a 2A linker is much more effective, whereas it's the opposite for EGFP Neo, where direct fusion works much better. And again, this is something you really need to test. We actually encode all this information within our vector design studio on our website. So if you are designing vectors and you want to know for your specific vector which combination works best, you can go on there, and the options we give you will be the ones that ensure you get the best expression levels of your fluorescent proteins as well as markers. So now let's move on to whether you should include a regulatory element or not. So inclusion of regulatory elements such as WPRE can actually significantly enhance transgene expression. But there are safety concerns associated with WPRE because it encodes an ORF, which is kind of linked potentially to oncogenesis. So for research applications, WPRE is generally fine, but for clinical development, it will really get through regulatory bodies if you include WPRE. Instead, it's better to include a modified version such as WPRE3, where this ORF has been mutated, so you don't have to have these worries. The location of WPRE actually also really affects gene expression of your upstream and downstream cassettes. So you can see here that you get sort of good expression of both your upstream and downstream cassette if you put WPRE last. But often your last cassette tends to be a fluorescent reporter, whereas your first cassette tends to be the gene of interest that you're really interested in. So we generally recommend that you would put your WPRE after your gene of interest. So importantly, backbone optimization can also really enhance production as well as scalability. And here we have different lentivirus constructs, and we're comparing different backbone versions. And you can see, depending on the backbone that you use, this can significantly affect both volume yield as well as specific yield. And this is really important from a clinical perspective if you want to scale up to produce really high titers of virus. Similarly, for clinical development, antibiotic choice is also super important. So for sort of foundational research, ampicillin is very commonly used, and we would recommend that. However, if you want to transition, if you're planning to take this vector for drug development purposes, we recommend that you switch it to kanamycin, otherwise it won't make it through regulatory bodies. And this is because ampicillin is a beta-lactam, which is a very commonly used type of antibiotic in medicine. And we want to avoid any resistance occurring to this type of antibiotic. Ideally, if you can not include an antibiotic for selection, this would be even better to increase safety for clinical development. And there are ways to do this. So you can use plasmids that use alternative selection mechanisms. For example, we have a miniaturized plasmid, which is called Minivec, and this allows both antibiotic and supplement-free selection, and this will facilitate much safer production. It also helps with manufacturability and cost because you're not having to use antibiotic for selection. So although the transfer vector is really important and we should carry out optimizations, it's also important to consider the other plasmids we're using as well for packaging. And so the envelope plasmid is actually really easy to modify to enhance your vector's efficacy in targeting certain cells and tissues. And as I said, we can easily pseudotype lentivirus by switching out this envelope protein for an envelope glycoprotein from another virus. VSVG is actually very commonly used to pseudotype lentivirus, and this is because it facilitates broad targeting of lots of different cell and tissue types. However, if you want to achieve more targeted delivery, then you can pseudotype with other viral envelope glycoproteins, for example, influenza A virus, which will facilitate targeting of the lungs. Another way that you can use pseudotyping is to study highly pathogenic viruses. So, for example, during the COVID-19 pandemic, SARS-CoV-2 envelope protein, you could pseudotype lentivirus with this to study things like entry mechanisms or test different drugs. And this meant that instead of having to use a biosafety level 3 facility, you could instead use a biosafety level 2 facility. And here is some data just showcasing this. So we've pseudotyped lentivirus with the spike protein of coronavirus, as well as different variants of the spike protein that emerged. And then we are transducing 293 T cells expressing the receptor ACE2. And you can see that depending on the variant of the spike protein that we used, we got variable levels of expression. So again, just to highlight really how this flexibility of pseudotyping can be used. So finally, let's move on to packaging optimization. So as we previously said, there are several different plasmids involved in packaging. This is, again, the 3rd generation system. So we have our transfer plasmid as well as our packaging plasmid, and we can optimize the packaging plasmids as well as optimize the packaging protocols. So we can play around with the types of promoter we use. We can play around with the different ratios of our packaging plasmids. And we can also optimize the protocol for packaging more like sort of trickier genes. So for example, toxic genes. And here we're showcasing some data where we are utilizing our optimized packaging protocol as well as our optimized packaging plasmids. So here again, we're using our miniaturized plasmid MiniVec, and using smaller plasmids in packaging can actually greatly increase packaging efficiency. And again, this has the added bonus of both antibiotic and supplement-free selection. And here you can see for our different constructs of different sizes that we are achieving compared to sort of lab systems or conventional second generation systems, and compared to other packaging systems from elsewhere, we can see this marked enhancement in packaging efficiency here shown in pink. And finally, I want to talk about packaging optimization for trickier genes. So as I said, for toxic genes. And toxic genes are basically any genes that may affect like the cell, the health of the cells that you're using for packaging, or the virus. So for example, types of genes that may affect the cell include cytotoxic genes, as well as pro-apoptotic genes and anti-proliferative genes. So for example, caspases. For inhibiting the virus, for lentivirus itself. This can be genes that would inhibit HIV packaging. So for example, ApoBec, SERINC3 and SERINC5. And we recommend the strategy to sort of overcome this is to minimize expression of your gene of interest in packaging cells, and then restore expression once it's in your target cells. And like we previously talked about, we could use an inducible promoter to do this, so something like the tetracycline system. And we just need to make sure that our transcriptional activator is present in our target cells. And and then, or we could use a tissue-specific promoter where we'll only see activity, so we'll only express our gene of interest when it is present in those target cells, and it will be inactive in the packaging cells, so it should not affect the packaging cells. So, in summary, if you want to carry out long-term expression in vitro and also for ex vivo therapy, then lentivirus is a great choice. It integrates into the host genome, and it can infect dividing and non-dividing cells, so it hangs about for a long time and allows long-term expression of that gene. And it does show promise for future in vivo therapies as well, although it's not quite there yet. But I think this is an exciting time for the use of lentivirus in different therapies. A rational vector design approach from the very beginning is crucial to enhance safety, efficacy, as well as manufacturability of lentiviral vectors. And this is both for research and clinical applications, and can save a significant amount of time and resources when you get really far down the line and then realize that your vector is not performing as it should. The good thing about lentivirus is it's super flexible to pseudotype. So depending on what you want to use it for, if you want to target different cells and tissues, you can switch out that viral envelope glycoprotein, which means that it's incredibly useful, again, both for research and clinical applications. And finally, to be able to make sure that you can achieve really high quality, high titer viruses, you can optimize the packaging process. And this can have huge effects on your titers. And again, this is really important for clinical development where you want to scale up and produce really high titers of virus. Yeah, and I would just like to thank everyone for attending. I've really enjoyed discussing all things lentivirus with you today. And if you're interested, in gaining more information or a bit of help with your vector design or lentivirus packaging, then yeah, please feel free to visit our website or contact us on services@vectorbuilder.com. Perfect.
Excellent. Thank you so
Excellent.
much, Skye.
Thank you so much.
That was wonderful. So we do have some questions, and I want to remind everybody that if you have questions, please type them in the little Q&A box, and we will get to them. Our first question is, do you have trouble with the TTS repressing the selection marker further downstream of the GOI?
Um, yes, let's go back. Is it not letting
So yeah, we also have oh
Let me just, I was
yeah,
just going to go back to the text and stuff. Yeah, so if we sort of have issues with the TTS inhibiting expression of the ORF, so you can see here that yes, when we include TTS, it can lead to sort of slightly reduced levels of expression of our ORF following doxycycline treatment. But I think it's really kind of a balance between making sure that you don't have that leaky expression and then making sure that you have good levels of expression of your gene of interest. And generally, if you're using a tetracycline inducible system, I'd say you're using that because you want to be able to control the timing of expression because you're expressing something like a toxic gene, for example. So yeah, I would say it really depends on what you're using this system for. And although it can slightly reduce expression of that ORF, it's probably a worthwhile payoff to make sure that you're not getting that leaky expression.
excellent. Our next question is, are there any selection markers for primary neurons or very delicate cells that probably cannot tolerate neo or puro?
Yeah, that's a good question. Um, so in terms of yeah, different selection markers. I guess, like personally, I haven't had much experience working with neurons, but I guess maybe you could use sort of like a fluorescent reporter instead if you're struggling, like you could use like flow cytometry or that sort of thing if you're struggling with finding an antibiotic that's not too strong and is not going to kill your cells. But again, I think that's something that you really have to personally test depending on your experimental setup. Like you really need to do sort of antibiotic kill curves and see and test different types of antibiotics. So sorry, there's not like a definitive answer, but I think it really depends on your experiment. Yeah.
Thank you. You mentioned ampicillin resistance constructs won't get through clinical approval. Isn't this resistance just for the E. coli used when producing plasmid? I thought that only Puro or Neo could be used for actual cell type selection once the virus is actually expressed in your cell of interest.
Yeah, that's a good question. Actually, Jen, do you mind stepping in for that one? I don't know if Jen.
Yes, yes, of course.
Perfect.
Can you hear me properly?
Yes.
Yeah.
Yes. So regarding this question, you're absolutely correct that ampicillin is used for E. coli selection, and we do recommend kanamycin instead of ampicillin to use to maintain the E. coli stocks. So both ampicillin, either ampicillin or kanamycin, they're part of the backbone components. They're also really important for downstream applications, especially if you want to go to, preclinical or clinical stage. And you're right, again, that puromycin, neomycin, those markers are usually used for selection in mammalian cells, for example, your target cells. So both elements are very important and they should both be carefully considered if you're planning to move into preclinical or clinical stage.
Excellent. Thank
Yeah.
you, Jen. Jen is our application scientist and absolutely wonderful. All right.
Yeah.
Our next question. Do you have any experience in transducing mesencephalic embryonic stem cells? If so, do you have suggestions on how the lentiviral vector should be designed for successful transduction? Jen, Skye, either of you can jump in.
Yeah, so personally, no, I don't have specific experience with that, but I think again, you can play around with different pseudotypes for embryonic stem cells. So things like RD114, envelope glycoproteins, as well as I think GALV as well, can transduce, I believe, can transduce these kinds of cell types. So I think, yeah, play around with the pseudotype of your lentivirus to try and achieve more targeted delivery. And again, you need to be considering things like your the promoter that you're using and all this kind of stuff. So again, for these types of cells, you need to stay away from CMV promoter. Um, and yeah, because it can be silenced and use different promoter types. So yeah.
Awesome. Thank you, Skye. Um, is it standard for lentiviral transgene plasmids to have low yields even if you use a high copy backbone? How do you fix this issue?
Um, again, it's so dependent on your construct. So yeah, again, I don't think there's like a definitive answer to this. So in terms of packaging efficiency, it can depend on so many things. So it can be really dependent on your um like transgene as well. So if you've got say like high GC content or you're trying to express toxigene or all this kind of stuff, so you need to really consider your transgene. In terms of like high copy number, I think that's more of an effect in the bacterial cells. So when you're growing up lots of copies of your transfer plasmid, the high copy number is more that you will get lots of copies produced in your bacterial cells. But for the packaging process, the things that you really want to more sort of focus on are your packaging process itself. So things like the ratio of packaging plasmids that you are using. Also, you can use miniaturized plasmids, like I said, that are smaller, like MiniVec, and these will lead to much higher transduction efficiency than we've seen that this, sorry, not transduction efficiencies, it will lead to much higher titer of virus from packaging. So it leads to much higher packaging efficiencies. So, so yeah, I think these are more the things you want to consider if you want to improve your packaging efficiency.
Wonderful, thank you. Our next question is, does the CAG packaging reduction also apply for other viral systems like AAVs, and is it mainly due to toxic overproduction of the transgenes in the packaging cells?
That's a very interesting question. Yeah, so I personally don't think that any of our data has pointed towards the CAG promoter effect in AAV packaging. It seems that I don't know, Jen, correct me if I'm wrong, but I think it's more been for lentivirus. And I believe it's because the CAG promoter has quite high GC content, which may affect packaging efficiency.
Yes, this is my experience as well. And then just adding one
Mhm.
point, the CAG CAG promoter is very effective in AAV and it's very commonly
Mhm.
used, especially for in vivo applications of AAV.
Yeah.
Yep.
Awesome, thank you. We have a whole lot of questions, and in the interest of time, we may not be able to get to all of them. We will maybe do a couple more questions, but please feel free to reach out to us at any time with your questions, and we will be more than happy to get back to you and answer them. We really
Yeah.
appreciate the enthusiasm and apologize that we don't have a whole lot of time. So maybe we can end it with this one question. I wanted to know what are the best methods and practices for viral titering, and how do these differ when you have reporter markers, that is GFP, versus when you only have selection markers, that is antibiotics, by themselves to determine the best MOI?
So I think for titer, there's a few different methods you can use. So P24 ELISA is often used where you're actually detecting the P24 protein on the surface of the virus. But obviously this detects all of the virus. It does all the viruses, even if they're not replication competent. So it gives you an idea, but it's not like super quantitative. You can also use things like qPCR. So again, this will measure the viral genomic RNA, or you can measure the integrated RNA. But again, this it can detect viruses that are not necessarily replication competent. But then if you have that fluorescent reporter, that can actually be really handy because then you can measure transduction efficiencies. So then you can transduce cells with your virus and see which cells get infected, because then you can detect your fluorescence via like FACS analysis or this kind of thing. So that's actually kind of better because then you can see the MY in terms of your actual infectious virus, instead of just all the virions, some which may be defective. So if you can include a fluorescent reporter, this actually makes titering much easier in determining the MOI that you want to use for your experiments. So, and again, for determining MOI for your experiments, I think you really need to just test different MOIs to see which ones will be the best for you.
Thank you
Did
so
you have
much.
some tips? Yeah.
All right. With that, I think we will conclude today's talk. And for the people that answered questions non-anonymously, we will be reaching out to you with the answers. I've made a note of all of it and really, really appreciate the enthusiasm. Thank you so much. And thank you, Skye. This was an excellent talk. And I hope that we see everybody in our next gene delivery seminar series in April. Thank you.